Hi, my name is Luke, and for this semester in our genetics class, we were tasked with researching and presenting a genetic disorder. So my genetic disorder I will be presenting to you today is maple syrup urine disease. Maple syrup urine disease is a disorder that blocks the body's ability to process certain amino acids through protein dysfunction. The amino acids in question are leucine, isoleucine, and valine, which outside of the body are common in milk, eggs, and meat. The buildup of these amino acids inside the body can cause toxicity to the organs, the brain, and the cells and tissue. The name of the disorder is from the distinctive symptom in which it makes secretions and excretions of the diagnosed have a sweet odor in reference to their urine, sweat, and earwax. So the prevalence with this disorder, it affects about 1 in every 185,000 infants worldwide. The testing for this can be done, as we will discuss later, through newborn testing. And a study done in California between 2005 and 2010 did show that there are different rates by ethnic backgrounds. So for the Middle Eastern background, about 14 in every 100,000 infants were found to have this disorder. For Filipino ancestry, about 2 in every 100,000. And then for Hispanics and other ethnic backgrounds, about 1 in every 100,000 were found to have it. It is worth noting through Medline, it was shown that the Mennonite population has the highest prevalence, with 1 in every 380 infants being diagnosed. So the symptoms and characteristics of the maple syrup urine disease can be categorized into 4 separate categories based on their age of onset, severity, and the enzyme activity. So the classic version, which is at birth, this one has about 2% or less enzyme activity. The intermittent is slightly different than the classic, just a little more severe, and its onset is 1 to 2 years old, and the enzyme activity can be between 8 and 15%. With the intermediate, this one, the age varies drastically, so there's no really set age, but the enzyme activity is about 3 to 8% of activity. And then the thymine responsive is present after infancy, and the enzyme activity in this one isn't really determined, but it is improved with the dosing of thiamine or vitamin B1 to the infant. So, as I stated, symptoms can vary based on the severity and type of disorder that they have, but it does show through Healthline that the older of the onset of the disorder, the worse the symptoms. So with the classic and intermittent, common symptoms are lethargy, poor appetite, weight loss, irritability, the distinctive sweet odor, the irregular sleeping patterns, and high-pitched crying. For the 2 later onset, as I said, with the worsening symptoms, the diagnosed can develop symptoms such as seizures, neurological deficiencies, developmental delays, feeding problems, poor growth, and the distinctive sweet odor in the secretions and excretions. So the maple syrup urine disease itself can affect 1 of 3 different genes. It can also affect multiple of these genes. The genes in question are used to encode the catalytic components of the branch chain alpha keto acid dehydrogenase complex, or the abbreviated BCKDC. When activated, this is what causes the component to break down the amino acids we discussed before, the leucine, isoleucine, and valine. The genes affected, as you can see, are part of that branch chain, and they're part of the alpha polypeptide in the beta subunit, as well as the dihydrolipomide branch chain transacrylase E2. So the normal properties of these genes are with the BCKDHA, it encodes part of the alpha subunit. The BCKDHB encodes part of the beta subunit on the branch chain, and then the DBT encodes part of the dehydrogenase complex as well. So when mutated, the BCKDHA it can show that the most common mutation is in the single alpha subunit amino acid. And furthermore, through Medline Plus, I did discover that within the Mennonite population, it was found that the most common change is the amino acid tyrosine to the amino acid asparagine at the position 438. For the BCK-DHB, the most common mutation was in the beta subunit. Medline found that within the Jewish community, the most common change in this one was arginine amino acid to proline amino acid at the 183rd position of the beta subunit. And then with the DBT, this one changes, it can change the DNA building blocks or the base pairs, and it can also cause insertion or deletion of portion of the DNA. All mutations with these three genes do cause the enzyme function and protein function to decline, and with this, it breaks down the body's ability to break down the 3 amino acids that we had discussed before, and the buildup of these amino acids within the body causes toxicity to the cells, tissue, organs, and the brain within the body. So maple syrup urine disease is a autosomal recessive disorder. As you can see, what I have put together here, the genotypes possible are the afflicted and carrier with the 2 lowercase m's. You have your genotype of the capital M, lowercase m, which is a person that's a carrier but not afflicted by the disorder. And then you have your 2 capital M's, which are the neither carrier nor afflicted. So with the parents that are a carrier or afflicted with the disorder, there is a chance that the child would also be a carrier or afflicted. And with parents that have neither carried or been afflicted by the disorder, there is a chance that the child may be the same and not be afflicted with the disorder or carry it. So I have put together a couple Punnett squares and affliction probability just to show the severity of this disorder. So with both parents being afflicted, there's a 100% chance that the offspring are going to be afflicted and carry the disease. With both parents being carriers, the children are a little more less susceptible. So 50% would be carriers, 25% would be afflicted and carry the disease, and then 25% would neither carry nor be afflicted by the disease. With one parent being afflicted and one parent being a carrier, on the other hand, 50% would be afflicted and carry the disease, and then 50% would just carry the disease. On the other hand, one parent being afflicted and one parent having neither affliction nor carrier, 100% of your children are going to be carriers in this instance, but none will be affected by the disease. With one parent being a carrier and one parent being neither, you'll have 50% of your offspring that carry the disease and 50% that are neither carriers nor afflicted. So I did put together a simple pedigree of a autosomal recessive disorder. As you can see on the sheet, it is determined to be very hard to eradicate from a family tree just because of the mode of inheritance. Only in an instance where one parent is neither a carrier nor afflicted is there a chance that the offspring may not be afflicted by the disorder. As you can see, just to help with the flow of my pedigree, I did eradicate several of the non-carriers just to show the inheritance and how rapidly it can spread. So with autosomal recessive, it is very hard to get rid of. So the testing for the genetic disorder itself, there are over 750 tests available for the genetic disorder, as you can see through ncbi.gov. Of the testing, the 2 major categories are either biochemical or molecular testing. The most common way of screening would be the newborn screening, which is a stick in the heel of the baby at birth to test for multiple disorders. The fee for the newborn screening does vary by state, but with me being from Kansas, I did my information based on Kansas, and it was through newstep.org. It is shown that Kansas provides free testing, and furthermore, if your child requires further testing, they will cover those expenses as well. It's not mandatory to test for this disorder, but left untested with the classic or intermittent, your symptoms will be onset fairly rapidly. One thing that is scary that I did notice is that a lot of the symptoms are common with colicky babies as well. So not doing the right treatment or not knowing exactly what your baby has can be detrimental to their development and future. It is shown through ncbi.gov that some children left untreated within 10 to 12 days can be comatose. So the earlier the testing done, the better the result would be in the long run. There are that I could find several ethical issues behind the newborn screening. Currently not, but there has been talks about integrating the testing with the whole genome sequencing, which would test the whole DNA sequence for disorders throughout the whole database. And one thing that I did find through NCBI is that this kind of testing would be overwhelming to both the healthcare provider and the parent of the testee. To elaborate on this, with the whole genome sequencing, they would be testing for literally everything. And the results could be overwhelming. And a lot of the results that you would be getting may not be relevant in the newborn, but may be relevant later in life. So to give all this information to somebody at one time can be overwhelming, and processing this information would be more difficult as well. So as you can see, or my works cited, I did quite a bit of research on the medlineplus.gov and the ncbi.gov as well as Healthline and omim.com. If you want to research further into this disorder, feel free to use my references, and I hope that I have answered some questions in regards to this disease. I know it's not completely prevalent in common knowledge, but anything that I can do to help, I surely do appreciate. Thank you for coming to my presentation.